Okay, great. Hi, everyone. Like Kelly said, my name is Kathryn Scafidi. I work with Dr. Mark Steele at Cal State Northridge, and my overall research has to do with studying the effects of an invasive alga on the overall trophic dynamics of temperate rocky reefs. And I'll get a little more into that down the road. But first, I'll start off with a little bit about me and how I came to be here. I was born and raised for my childhood, at least, in Maryland, and I grew up kind of going to the beach and digging through the sand and interacting with the ocean life. And then I moved to Northern California, where my parents and my two perfect dogs reside. That's not biased. They're the best in the world. And so I was very landlocked when I was up there, but I still had a heavy influence with the ocean life because we would visit Southern California every year. And so I originally had gone to school for photography, and I got dive certified because I wanted to work for National Geographic. So this kind of all ties in because it's a funny path that I didn't actually have it in my mind to be a marine biologist, but I had the skill set of being dive certified, which came in handy later on. And so I really found my passion for marine biology when I took a couple of courses at a community college, and we'd go out in the field, and I learned about the intertidal. And then I learned about some of the more deep water organisms, but I hadn't really been diving in a long time. And so I decided to do marine biology, where I transferred to Long Beach State, and I pursued my degree there. And it really took off because I got to spend a semester out here at the Wrigley Institute taking courses, and that all went towards my bachelor's degree. And I actually met my advisor out here because he was one of the instructors. So then I decided to go to CSUN for my graduate degree, where I'm currently doing a master's, and I've worked on projects. We work on projects off the coast of San Clemente, and we're working on projects with the San Onofre power plant and looking at the artificial reef that they have out there and the natural reef to see how that mitigation is going. And we've also come across, this is a giant black sea bass. This is actually a juvenile, which was insane to find one of these out in the water. So we've been doing some spot analysis with these guys to see if they're frequent visitors of these reefs. And then I also spent a summer out here before I started working for my advisor on a project where we looked at these two sex-changing fish. They're gobies, the black-eyed goby on the left and the blue-banded goby on the right. And so then I got into my own project where I'm looking at invasive species and how that plays into our environment. And so this map here shows there's all these white lines are pathways for those big freighter ships that come into the port of LA. So as you can see, there's almost no coastline untouched by these pathways. And so what happens with these is these freighter ships are capable of carrying these different organisms across natural barriers, so like the open ocean. They're able to carry organisms from one coastline, like over here around Japan, all the way over to, say, us down in LA. And so how they do that is they have this ballast system. So if we look at this big freighter ship, it's empty of cargo on the top there. And underneath here are these sections that are down in the ballast. And that water that they fill it with is called ballast water. And they fill it at one port when it's empty, so it'll keep it weighted down as it travels to its destination port. And so if we look at these triangles and circles and pretend that those are, say, just species of algae, or they could be larval fish, things like that, they get trapped into this ballast water. Then they can hitchhike their way all the way across the open ocean. And when they get to their destination port and they bring on the load that they are going to carry somewhere else, they offload that ballast water, and then those species are now in the water at those other destinations. And so oftentimes these are obviously like in harbors and coastal habitats. So some of these species are capable of establishing themselves, which then makes them really good invasives because then they can persist in this new environment. So that's the main reason, or the main way that these marine invasive species can spread. So again, if we look at this map with the pathways and we zoom in over here to Southern California, mainly the port of LA. That is one of the largest ports in the US. So we have over, I think last year they had over 1.4 million ships that had visited the port. So that's a lot. And so that obviously makes it super susceptible to these invasions. And one such invasive species is known as Sarcasm horneri. It's an alga or seaweed. And so this alga is native to South Korea, Japan, and China. So it's been brought over by one of those freighter ships. It's known as an annual brown alga. So that means that it completes its life cycle within a year. So it's able to reproduce and die within a year, but it can do that every year, over and over. It's this is a technical term, monoecious, which means that it is capable of reproducing with itself. So it has both sexual parts on one organism. So even if it's detached and it's drifting away, it can float, it can still reproduce. So that's how this spreads, is that even though, unlike plants, if you uproot it, it'll die, these can still live and photosynthesize as they float along, and this one specifically can reproduce with itself. And then they have these other things known as secondary metabolites. So essentially, it has these chemicals that it can excrete that makes it unpalatable or distasteful to other animals that are trying to eat it. So that's actually a really good preventative measure to protect itself as it spreads. Get back to that later on in my research. So here we are over, if we look down here over in Los Angeles, we have the Southern California Bight right here in the Northern Channel Islands and Southern Channel Islands. A study was done in 2015, it was published, but it was over the course of about 10 years. They were looking at the spread of this alga. It was first discovered in Long Beach Harbor in 2003, and then it was found here on the leeward side of Catalina in 2006. So if we look here, this is Catalina circled, and then there's the Port of LA, Long Beach Harbor, where it was discovered. And so this was its initial kind of, people had reported that it was on the leeward side here. There's one spot down here in San Diego. And then if we just kind of go through this time series, you can see very quickly it starts to spread from the northern part of the Southern California Bight all the way down to San Diego, and actually it's now invasive down into Baja. So over the span of 10 years, it's rapidly spread. And these are actually, these could also be, these are obviously, people have reported this. There are other islands that are very accessible to the general public, and if research isn't going on, it could also be there. So bear that in mind. It could be in more spots that people just haven't seen it yet. So my overall question for my research that I'm doing right now is, what effects does this alga have on multiple trophic levels in our ecosystem? And to just go over trophic levels and trophic interactions, what I mean by that is looking at an overall food web. So what's going on? We have our primary producers, so say like grass, that is photosynthesizing. It's providing the base food level for other organisms as we work our way up. And so we also have primary consumers like this grasshopper or other insects that eat the grass, and they get nutrients from that. Then we have secondary consumers, so they're eating the grasshoppers or the insects, like this mouse. And then we have our tertiary consumer, which would be like our apex predators, so our eagles, wolves, bears, big animals that are consuming other animals. So we have this kind of chain reaction of events going on here, working our way up through this food chain. And so if we look at in the marine system here, I have our primary producer would be an algae of some sort. We have the primary consumers would be small little invertebrates, so like very, very small crustaceans, snails, clams, things like that, that live among the algae and kind of forage on it slowly. And then we have the secondary consumer, which could be like most of the reef fish you'll see out here that eat these small little inverts. And then if we look at the tertiary consumer, it would be like the big fish, say like a big bass of some sort. But for my research, I'm just looking at these first three levels. And so what I want to know is what is going on with the indirect effects. So what's going on from the primary producer level all the way up to the secondary consumer level, and how that whole system being affected in the event that there's an invasive species, such as Sarcasm horneri. So what I started off with is fish foraging observations, and then I want to look at the invertebrate epifaunal abundance, so the organisms living on the algae, and then I'm coming back to those phenolic compounds of secondary metabolites. I want to understand if there is a difference in the concentrations of those between the invasive and the natives. And what I expected to see was that there would be lower foraging among the invasive because the fish aren't familiar with it and they kind of actively avoid it, and that would be due to there's just no food there for them. So these invertebrates are not living among the invasive, and that is because the invasive has these high concentrations of these chemicals that make it undesirable for them to eat. So this is kind of my thinking of what I was going to see with my research. And so my study sites are up here off the leeward side of Catalina Island. We're over here at the Wrigley Institute. I have 6 sites ranging from down here. This is Empire Landing and all the way up to Arrow Point. I have been, so I started off with foraging observations that I did last year. I'm not continuing that this summer, but what I looked at were 3 of our common reef fishes. So we have rock wrasse at the top, we have California sheephead and Garibaldi. And I picked these guys because they all forage among the algae in a similar manner, in the same depth area, and they all kind of forage among all the different species of algae. So I could get a better representation of, are all the fish interacting with it equally? I then did these benthic surveys where I went out and tried to quantify the percent coverage of all the algal species in these areas that I did these observations. So I could say that these were the observations I saw. The fish were eating among these species, and this was what was available to them. So if I saw that they were foraging among a certain native alga a lot, but say its percent coverage was really low, they're actively choosing that compared to if the invasive was mainly dominating the reef, that they're still actively avoiding that. So that would kind of present a better picture of what they had available. And then I've done these algal collections with, this is the invasive Sarcasm horneri. We have 3 native species, Sarcasm pomeri, Dictyopteris undulata, and Zenaria farlowii, and all 4 of these are brown algae, so they have those phenolic compounds. So I can compare the invasive with 3 of the most abundant native species and see if I can find a trend with that. And I collected these in these tea bags that have a really fine mesh, so I could collect algal individuals, and all the invertebrates that were living on the algae would be collected at the same time, and I could take it back to the lab and count them and identify them under a microscope. And then with those algal samples, now that they're cleaned of the inverts, I then can extract their phenols and run those to see what their concentrations are. So I'm going to throw up just a couple graphs here. And these are just based on what I found so far. Again, the foraging, I have completed that section. And so right here on this y-axis, I have the foraging rate, which is their number of bites of individual fish took per minute. And I did these in 5-minute time surveys. And then down here are the 4 species of algae. So these first 3 are going to be, this will be the same on the next graph. These are the native species, and again, the invasive is all the way on the right. And if we look at this, you can ignore the stats there, but if you look at this, there is a dramatic difference in foraging among the native Sargassum species compared to the invasive. So that tells me that the fish are actively avoiding it, and potentially it's due to not finding food on it, but this kind of solidified my hypothesis that they would not forage among it as frequently because they have these other choices of the native species. And between Dictyopteris and horneri, one of the natives, and the invasive, there is not quite a statistical difference, but you can still see an overall trend here of how often they were foraging among the native species versus the invasive. So then I want to look at the epifaunal or the invertebrate epifaunal community and see if are there none on the invasive, or maybe there's a lot and the fish aren't eating them, So again, over here on the y-axis, I have epifaunal abundance, so just how many of these little invertebrates are on the individual algal samples. And then I have my algal species on the x-axis. And that told me, like, that's the reason why. There really is not a whole lot of these, of this food source for the fishes living among the invasive species. So that was that solidified to me that there's something going on, and this could be maybe at that chemical level, which are those phenols that I'm looking at. So I don't have my data for my phenols done yet. I'm still extracting the chemicals, and I'm still going through some more samples of the invertebrate abundance. But so stay tuned. Maybe next year, if you see me at another conference, I'll have my data. But looking at the overall, what I've gotten so far is, again, I looked at the fish foraging, and it was lower among the invasive, and the invertebrate abundance was also lower. So I'm going to look at the total phenols and see if this could be the reason why. Maybe it's deterring that. So that's kind of to be continued. And then I'm also looking at, I don't want to overwhelm you guys with too much, but I'm doing fish collections of rock wrasse specifically, where I'm looking at their growth and their reproduction to see if that changes in these different sites with various densities of the invasive. So maybe it's affecting them. And if they're not getting enough food, then that could be potentially affecting them down the road too. So that's kind of what I'm doing this summer, is working through these samples and getting more fish collections. But overall, so why do we care about this? Why do I care? Why do you guys care? Why should anyone care? So if this invasive does not provide the habitat for these prey for fishes, it's important to identify that, because we need to make the efforts to try to figure out how to mitigate these effects. So if fish aren't getting a proper food source, maybe like down the road, seasonal fishing could be a way to kind of help fish recover from that fishing pressure, but also not having the resources in their habitat to actually grow up to the size limits that we have in place. And then I think that studies overall for these different ecosystems, mainly like our reef ecosystems, it's important to understand the overall trophic interactions that are happening in these ecosystems, because then we could potentially identify their susceptibility to invasion. So some ecosystems have been found to be a little bit more resilient to that, which is great. So it's important to study those interactions and see what's going on there, how that's working. And other ones that could be more susceptible to this, we could put into effect certain measures to try to help keep that from happening. And then invasive species research can also inform our policymakers on ways to prevent future invasions. So, active boaters that come out here from the mainland to this island, and vice versa if you're traveling around, making sure that you don't carry an invasive species with you unintentionally, or finding alternative ways for those big freighter ships to prevent carrying invasive species. And one of those ways is that they can offload that ballast water in the middle of the deep ocean and then uptake it again, because things that are found on the coast cannot survive in the deep ocean, and then vice versa. So if you uptake water from the deep ocean and bring that into your port, most likely any of those things that are floating around in the water there aren't going to be able to survive. So that's kind of a simple way to prevent that. And these are all the people that have helped me. It takes, a village to try to get your graduate work done, so it's great, and funding resources, and especially the Wrigley Institute, where I've been able to do all of my diving and my research out of. And I'll take any questions. Yes.
What type of range do the reef fish that you were looking at typically have? Like their
home range of swimming around?
Yeah.
Yeah, so that's a good question. Our reefs along the coastline here are they're large. They have a large length, but not too deep of a reef where these fish and these algae are found.
Yeah.
But then these different sites are separated usually by a large sandy bottom habitat, so they tend to not travel like from, say, just right outside our cove here at intakes, the intake pipes here, they don't tend to like travel over that open channel to the isthmus reef. They'll kind of have a very close home range. So my areas are about 200 meters long, and then I did my surveys between 10 and 30 feet, kind of account for them traveling around. But yeah, I would say that the Garibaldi are they're very territorial, so they have smaller home ranges that they keep track of, and the rock wrasse have a slightly larger, but they're still very, I would say it's probably like 10 meters around. They'll kind of hover around and hang out at. And sheephead are a little more mobile, but the ones that I was looking at were smaller sheephead, because when they get larger, their diet shifts, so they eat larger prey like urchins. But the smaller ones have that same general small home range at these sites. Yeah.
Yeah.
Any other questions? Yes.
Did you select the sites by the pervasiveness of this invasive species, or just because these were the best sites surrounding a specific island?
That's a good question. I had some previous data from some lab mates that had been working on similar projects, and I selected most of my western sites like Arrow Point and Howland's Landing. I selected those because of their recruitment data, so they had seen how many of the young juvenile Sarcasm horneri were growing in the time that they were out here. And then some of the other ones I based off my personal observation, because when I was out here as an undergrad for a semester, this was kind of like the basis of that project that I did out here. So I had personal observation that I saw it was covering these reefs. So yeah, it was kind of like that, and I wanted to get a wide enough spread along the leeward side, because this side of the island has had it for the longest. It's been invasive here for the longest. So yeah. Any other questions?
Yeah.
Okay.
I know this falls outside of your specific research focus, but around this area, what would be some examples of apex predators?
You have sharks, you have the more pelagic species, but sometimes they'll come in a little close to the coastlines at different times of the year. So you have yellowtail, you have large bass, even kelp bass are actually, I would consider, maybe not apex, but like a tertiary consumer, because they eat other fish. So their main diet is typically other fish, but all of these fish eat, fish are kind of, they're opportunists, so they'll eat anything they can get their mouth around. So most of these guys, when they're small, when they're juveniles, they can only eat the very small prey. So I think it's important that we can identify where this comes from, because that's going to be their kind of basis until they can grow to eat other big fish. But yeah, and then sea lions, they're at the top of the food chain around here, unless a great white comes, skulking through. But yeah, those are some of the examples.
Have you seen the algae have any more effects on any other plant organisms around the area in California, or any other types of algae, like the native species?
Yeah, so it's kind of funny. The life cycle of this invasive is very different compared to, say, a lot of people have done a lot of research on how it's affecting giant kelp, macrocystis, because during the El Nino event, all the kelp essentially died off from the island because the water got too warm. And then this started to just kind of take off, because it's believed now that it's just because it's more of an understory alga, that the kelp wasn't shading it, so it was able to get enough nutrients and grow. But this invasive during the winter months and into actually just a month ago, it'll be taller than me at depths really deep, or even shallow. It kind of took over the cove for a bit, but then it'll die off around this time, and then we start to see little juveniles starting to grow again. So that's kind of its life cycle. So it will outcompete native species if there's space for it. But from what I saw when I did my undergraduate research out here, where it was just covering the reefs, now it's actually kind of started to blend in a little more. It's a little bit more interspersed with the native species. So I don't think it has quite the effect that everyone thought it was going to, because with macrocystis as well, the giant kelp, it kind of has an alternating life cycle. So when kelp is at its like peak growing season, this is pretty much died off / it's just the small little juveniles. So they're not really directly competing with each other. As far as some of the soft corals, I don't know any information by the soft coral like population around here, but that hasn't really been done before. So I don't know if it's actually directly competing with that. But a lot of these things, it's just space. Is there space for it to settle and grow? And then, sometimes there are like chemical interactions that could be affecting it. But right now, it's actually fairly mellow compared to a couple years ago. It was everywhere. Yes.
What are some ways we can restore the balance with a native species?
That's a good question. So they have, I think they still have an effect. There was years ago, it was just recreational divers could collect X amount of this invasive. So it was try to just take it off yourself from the reefs and get rid of it. That's unfortunate because like I said, if it floats away, it can still reproduce. So sometimes almost like anchoring and then dislodging it, it can still drift and reproduce and things like that. So you have to be really careful that you're not, getting any of the pieces, missing any of them and they go drifting off type of thing. As far as trying to help the native species with it, unfortunately for this invasion event, we're beyond the point of no return of actually eradicating this because it's covering too many places. It's everywhere. So now what we can do is only try to see how it's interacting with other things and see if we kind of can, meet it head on if we see these negative effects, or like I said, if it's starting to kind of integrate itself into the environment, and if it is able to provide this other habitat, it's more structure to the reef, so maybe things can live on it and everything. What I found is that's not quite the case, but I'm still trying to figure out what that could be, and maybe it's just a time series that it's going to take time for things to fully integrate with it. We had another invasive species, Sarcasm muticum, in the 50s, and that one is pretty much kind of integrated into the ecosystem and hasn't had a huge detrimental effect that we know of on the environment. So it could just take time. But when you identify these invasion events early on, there is potential to completely get rid of it. They did that in a lagoon off the mainland, and there was a green alga that came in and it was invasive and taking over. It's called Caulerpa taxifolia. I don't know if anyone's heard it, but that was one of the few successes of just completely identifying it and then quickly acting to get rid of it. But it gets expensive the longer it has been able to establish itself.
How do you know if it's more favorable to wait for the ecosystem to adapt to the invasive species or to just remove the invasive species? How do you know which one to do when you identify one?
That's a good question. Usually when people identify it, nowadays we have so many people that are active recreational divers, which is awesome. And there's apps like iNaturalist and things like that where people can post stuff and say hey, what's this? I don't know what this is. And people can identify it. So that's a cool way to actually track these invasion events. Again, it's kind of there's a bell curve figure for the invasion events. And so there's a point where it's going to be too costly to completely eradicate it. So at that point, you just have to keep monitoring it and making sure that it's not having this ripple effect to other species in the ecosystem. And I mean, there's a huge world of invasion biology where people try to figure out and counteract these events. And so at some point, it's going to be too expensive or just unfeasible to completely get rid of it. And that's why you need to just kind of see how it's doing over time and maybe try to help protect other things that are interacting with it in the environment to mitigate those effects. But early on, the earliest people can report something new and figure it out, the better. It drastically changes quickly, but if you can identify things early on, that's why it's kind of good. When you guys come into these different talks and stuff, and you can know what's native around here, and then if you're out snorkeling or something, and you see something weird, you can snap a picture, and that helps scientists everywhere to kind of keep track of what's going on. Any other questions?
In the 4 species of algae that you looked at, did you map their community, or could you anecdotally describe like how they mix and how big they grow?
Yeah, I didn't map it out per se. I kind of had like my surveys were in an area to kind of generally understand what was present, but they are actually fairly well amongst each other. Some reefs have like maybe on a bigger boulder there will be a giant patch of Dictyopterus or a giant patch of Sarcasm palmeri, the native one. And then Zenaria farlowii, it starts out as almost like this encrusting, it just looks like slime on a rock. And then as it grows, it'll start to change and you get that more leafy look to it. And so that'll be kind of closely related with the other ones, but they're all fairly well interspersed with each other in the reef. But those 3 natives are like the most abundant native brown algae that you'll see around here. So you'll see that with a lot of kind of the red algae. This is a lot of red algae here, but this is palmeri, and then there's Dictyopterus down here, and then Zenaria is down in there. But this picture, you can see more of the palmeri because that one will start to grow a little bit taller. Zenaria is a little shorter, and then Dictyopterus is kind of in between.
How did you manage that while you were counting the bites per minute?
Looking at it,
Yeah.
or so I had to follow a fish around, and so I would start my stopwatch for 5 minutes, and I'd swim around and follow them at a distance that I wouldn't affect their behavior, but I could still see, and I would, I had to know all the algae that was there, or take notes if I didn't know what I was looking at, and come back and ID it. So then I could see that they took a bite on this specific species, and I just jot it down.
So you were counting the bite and the species.
Yeah.
But were those 3 fish feeding directly on the algae, or were they feeding on the invertebrates?
Yeah, so they take a bite and it looks like it's on the algae, but they're actually going for the invertebrates.
But the invertebrates are sessile on the algae. They're not.
Some of them are mobile. So some of them could be moving around different ones, but typically they'll kind of hang out with one alga. But there are little snails or clams that I found on some, and those are more sessile to certain species. Yeah, and so that's why I knew the algae that they would intake is more byproduct of their bite, trying to get the inverts. But so the fish most likely aren't being deterred by the phenols, the chemical compounds of the algae itself, because it's not strong enough in the bite that they're taking.
According to your hypothesis.
According to my hypothesis. But from what I found is, Garibaldi were the ones that were fighting off of horneri. So the bites that I got from the invasive were Garibaldi, and they're
So David Attenborough told me that Garibaldi feed off they garden their own algae. Are they eating invertebrates that grow off their garden, or are they eating algae directly?
So they're omnivores.
Or
So they'll eat a little. No. They'll eat a little bit of algae, but they're not. There's other. There's fish called opali that you guys will be able to see in the tanks that we have up here, and those are herbivores. So their main diet is algae. They're going around and feeding off of algae. I didn't pick them for this study. I wanted to, but their behavior is just all over the place. So I was okay, guys. But most likely they're getting more of their nutrients from these inverts if they can get it. So that's going to be kind of an easier prey to get with a higher benefit. So they're going to get more energy from that. But it does look like they're eating off of, and the inverts are also kind of eating off of epiphytic algae. So algae growing on algae, which is small and terpy. There's a whole lot of interactions going on here, which is what I'm trying to figure out. Are the chemicals actually affecting those guys directly, or is there another step in this whole thing? Mm-hmm. It's a lot, but all these fish will kind of get some algae intake, but they're not specifically algae eaters. They are mainly trying to get that higher caloric intake from the inverts.
Interesting.
You have a question?
Yeah, so you noticed the reduced feeding on the horneri. In your observations, did you see any reduced feeding on surrounding algae by proxy? Was there any reduced, how are the trophic levels on, I guess, a good algae that they enjoy that was directly next to a large population of horneri?
Yeah, it seemed fine. So the proximity of the invasive to a native alga didn't deter them from foraging off the native, and especially Pomeroy is out of all of the species, and it's kind of its morphology, so its shape of the branches and everything. It's got a lot of spaces in there. Closely, the branches are closely clustered, so there's a lot of spaces that the inverts can hide in there and kind of avoid herbivory. Or not herbivorous, sorry, predators. And so that alga in particular had a ton of inverts that I'd get off of it. And but it could be right next to horneri, and the fish they would cruise up to horneri and look at it for a little bit. Kind of, you can see them looking and trying to track something, and then they would go to the next alga, and they would like take a lot of bites off of that. Some fish it seems, this is not a, proven scientific fact, but my observations, it looked almost like some fish had different preferences. Like if one foraged among Xenaria a lot, it tended to take a lot of bites off of that. And then this one maybe like Dictyopterus. The prey are a little different. So there's more crustaceans and like shrimp-like things on Pomeroy versus like the snails and kind of clam things that you find on Xenaria or Dictyopterus. So that could be why. I don't really know about that, but the proximity of the invasive and the native didn't have an effect on their foraging. They just didn't choose that. And when I do my collections of the invasive, I can even just, before I go to count them, I have all my inverts in sample cups, and there's like barely any in any of my samples of horneri, even the adult ones that I got. And there's more branching and like more habitat for these things, and they're just not there. So I'm going to try to figure out, run an experiment this summer as well to see if there's a difference in these organisms visiting the alga and like living there long term, or if they're just not staying there. Yeah.
Let's go to Japan and look at the horneri there.
Yeah, that would be like my end all, on a master's budget. It's a little hard, but yeah, I would love to see how things are associated with it in its native habitat. Yeah.
And this will be the last question.
Yeah.
The process that you were describing with the ships, like the potential implications of getting them to offload in the deep water and uptake other water, how long would that take?
For them to switch out the water?
Is that like an easy, feasible thing for a company to do? Or?
That's a good question. Yeah, I don't know the duration of it, but I think some do it. So they are doing it.
Like
it's just not everyone.
Got it.
But I think what they can do is, and also you can't just dump it all out at the same time because then it'll tip over. But I think they can, like as they're offloading one, and then start to like cycle through water to fill. I don't know quite the mechanics
but in
of
practice,
it.
there are companies that are doing it.
Yeah, there are some.
Majority.
Yeah,
okay.
It's just like how do you put a regulation on that? That's like a global regulation that would have to be done. But there are some.
And that's international waters, right?
International waters.
Okay.
Yeah, and it's more of up to the captains from what I've found out and read, because I tried to figure out like what's going on with this. What are the ways that we can fix this? And it's kind of up to the captain's discretion. So there's certain captains that are like, yeah, I can do that, no big deal. And then maybe others just like don't want to deal with it. Or I don't know if there's a difference in like the ship mechanics and maybe like older ones versus new. Those are a lot of details that, yeah, it'd be good to look into. But there are some that are doing it, and that helps a lot for sure.
Thank you.
Thank you.